Students, further we will study about the sporogenesis in Riccia. Sporogenesis. It is the process leading to the transformation of the diploid protoplast of the sporocyte, that is the spore mother cell, into an haploid dispersal unit, the spore, with a resistance wall. The spore consists of a complex integrated series of events involving a special kind of nuclear division called meiosis. In meiosis, the diploid nucleus of the spore mother cell undergoes two successive nuclear division, but the chromosomes are replicated only once. In Riccia, almost all the cell of the sporogenous tissue differentiated into spore mother cell or sporocyte. As the sporocyte begins to separate from one another, they become more or less rounded. Their wall becomes mucilaginous and the content densely granular. Each sporocyte has a diploid nucleus. With the degeneration of their wall, spore mother cell increase in size and the diploid nucleus undergoes meiosis. The first meiotic division is reductional. It reduces the chromosome number to the half of the somatic number. The second meiotic division, which follows the first, is equational mitosis. Both constitute meiosis. The resultant four haploid daughter nuclei migrate to the periphery of the sporocyte and lie at equal distance from each other. The cell wall delimiting the four haploid spores are laid down simultaneously between the four nuclei. At the end of the second division, this group of four cells is called an immature spore tetrad. The cells in the tetrad are typically arranged in a tetrahedral manner, and the wall that separates them is in the form of a thin membrane. The four cells of the immature tetrad are enclosed at first in a common sheath. Each cell of the tetrad now secretes around it within its thin membrane a comparatively thick homogeneous callose layer. It is deposited unevenly around the spore protoplast and acts as a mold against which the marking of the mature spore coat are cast. Inside the callose layer is a deposit the first or the outer layer of the spore coat known as the exosporium in the form of a series of lamellae. The later project into and follow the counter of the callus layer. The second layer of the spore coat, which is known as mesosporium, is deposited on the inner layer of the exosporium. During the concluding stage of the spore development is formed the third and the innermost layer of the spore coat. It is termed endosporium. The three layers of the spore coat thus develop in a centripetal succession. As the spore matured, the spore coat thick considerably. The exosporium, which is yellow at first, changes to orange and become black at maturity. The callose layer and the sheath of the spore mother cell, which enclose the four spore, disappear. Consequently, the spore separates and lie free in the cavity surrounded by the outer layer of the calyptra. Sporophyte. The sporogonium of Riccia is the simplest among the liverwurst. It lacks both foot and seta. It is just a spore sac or a capsule spherical in outline. It has a wall 1 cell in thickness. The capsule wall encloses a mass of spore mother cell. The two-layered calyptra form a close investment around the sporogonium. The latter never emerge out of it. Before the spore mother cell divide to form spores, the single layer wall of the sporogonium disintegrate. Later, the inner layer of the calyptra also break down. The mature spores lie free in the cavity and a sac surrounded by the outer layer of the calyptra. There are no elaters. The mature sporogonium of Riccia has no diploid or sporophytic structure at this stage. The meiospore, which represent the future gametophyte, are housed in a sac provided by the parent gametophyte. Nutrition. Unlike the other liverwursts, the embryo, sporogonium and the spore mother cell in Riccia develop no chloroplast. Thus no starch is formed. The sporophyte remain totally dependent upon the thallus, that is the gametophyte, for the food material, water and mineral solution. Digestion of the sporogonium. The sporogonium of Riccia never dies. The spores are liberated by the decay or shriveling of the surrounding outer layer of the calyptra, and the thallus tissue thus perishes and dry. The spore remain behind on the soil. In this condition, they may be dispersed by wind. They remain alive for some time. Finally, they germinate with the onset of the condition favorable for the growth. The external morphology of the spores. The liberated meiospores is a pyramidal in shape. It consists of a tiny mass of cytoplasm containing a small haploid nucleus. The food is largely stored in the form of oil globule. The protected spore wall, which is known as the sporoderm, is thick due to the presence of black, and it is a thick, black and sculptured. The surface sculpturing varies from species to species. It is, however, specific and help to identify the species. The internal structure of the sporoderm is largely similar. The sporoderm, the spore coat, is usually differentiated into two layers. The exine and the inner is the intine. The intine is thin, about 0.3 micrometer in thickness. It is a homogeneous and composed of pectose and cellulose. The exine contains material to acidolysis. It is known as the sporopollenin. The major surface sculpturing of the spore comprises ridges and depression in the areolated or irregular configuration. Finer surface features such as tubercle or papillae ornament the ridges or depressions. The exine is thick and differentiated into three concentric, morphologically different lamellae regions. The innermost layer, or the layer of the exine, which is direct contact with the intine, is composed of thin, closely spaced lamellae. Spore germination. The spore germinate when it gets moisture. The spores germinate, the exospores come out, burst, and the intine come out in the form of a germ tube. It divides by transverse division to form the rhizoidal cell. Then further there is a division in the germ tube to form a large mass of cell, which later on differentiate to form the thallus. Thank you.